Hello, and this is, I don't know if you saw my last talk, but this is a project I haven't mentioned yet. We're developing a new citizen science project called beta catchers, and so I wanted to tell you a little about an issue we ran into in developing the project and how we're trying to resolve this issue, just because it seems like an interesting case study, and we're all in a similar field, and maybe we can benefit from learning from each other's mistakes. So sharing one of our mistakes. So the biomedical issue that we're tackling here is related to health disparity in Alzheimer's disease. So it turns out that Hispanics, I think in the US, are about 50% more likely to develop Alzheimer's disease than non-Hispanic whites. Biomedical researchers we're collaborating with are looking at both social and biological determinants, but this project is about the biological determinants. And so they're asking us to crowdsource the histopathological analysis of brain tissue samples taken from deceased Alzheimer's patients. They take a slice of brain tissue from someone who died of Alzheimer's disease, and then they take a very high-resolution microscopic digital image. And then our job is to analyze that image and try to, as citizen science participants, identify very specific kinds of lesions, in particular cord plaques, which look kind of like a bullseye with a little dot in the middle. And then these CAAs, which are really kind of cross sections of blood vessels that have absorbed or been saturated with the amyloid protein. These diffuse plaques we ignore in the citizen science analysis because we can use automated methods to easily just count the red stained pixels on those. So there's actually two components to the analysis. One is to identify these lesions, and the other is to quantify the burden of that particular lesion. So we roughly describe these as megatiles as the identification and classification task, and then mini-tiles, which is measuring how much there is. But for this brief session, I'm not going to talk about the mini-tiles. That one actually works great. Citizen scientists do a fantastic job of that part. You can see it's just basically drawing dots around an object and measuring it. But the megatile analysis is a bit trickier. So this is a screenshot from our pilot platform. And so basically, we let people sort of decide which kind of lesion they think they've identified. So you click on a cord button if you see a cord plaque, and then you make a little dot to indicate that you found it there. And then you can do that for CAAs as well. And here you see somebody who's already identified a bunch of things, and then they say next, and we have feedback because we already have an expert response that's previously analyzed this megatile, and that's the green one. So there's a little bit of overlap, but this is part of the training process by which we show what we call calibration examples, and then people learn by trial and error. So the issue is that normally a trained pathologist does this analysis. And the reason we have to crowdsource this is because there's such a high volume of data coming out of this project that the cost and time to get a bunch of trained pathologists to do this would be completely prohibitive. The problem is how can we trust the analysis of an untrained member of the general public to do this? And so we appeal to this wisdom of crowds approach that we take in our other projects. And so the question here is, if we have a bunch of things that look like this, which are only 25% correct, how do we combine them to get something that's almost 100% correct, at least 95% of the time? So we came up with this wisdom of crowds method that we call successive collapsing. And the simple idea is that if you take a bunch of megatile annotations from several different volunteers, imagine that each color represents a different volunteer, then we can use a proximity metric, and we can say, okay, if things are within this kind of, I don't know, oh, you can't really see my pointer. So yeah, you can see there's a distance threshold there. If the blue and the red labels are close enough together within that distance threshold, then we'll combine them into a single purple one that's a little bigger. So we assign a slightly heavier weight to that since it was two people saying the same thing. And then we center it on the distance between the red and the blue one. So now it's close enough to the orange one because it's been sort of centered that way, that's also within the distance threshold. and we combine the purple and the orange, and we have the green one. But the yellow one, and whatever you want to call this other color, they're too far away, so those remain distinct. We applied this method, and we piloted it to see if it would work compared to experts. And this is just an example of one megatile where we had a crowd answer. So the purple represents the combination of maybe 5 different people. So the big one means probably all 5 people thought that was a CAA, but you can see there's no expert answer there, so the crowd got that one wrong. The 2 near the bottom here, the crowd and the experts all agree. And the top one, the crowd agrees with one expert, but the experts don't agree with each other, which is always kind of interesting. So we ran an experiment, and we wanted to see, depending on how many people are contributing answers that are getting combined together, how does that compare to the expert answer? So if we have only one person answering using the F-score metric, we see it doesn't really align very well with experts at all. But then, as you have larger and larger groups, you get closer and closer agreement with the experts. So that top green line, that's how much the experts agree with each other. And our idea is if we can get the crowd to agree with either expert at least as much as the experts agree with each other, then we've achieved sufficient level of performance. But you can see that with 30 people responding about the very same megatile, we're getting close, but we're still not at that expert level of agreement. In fact, here projected out, it would take about 50 people. That's a lot of people looking at the same thing, not very efficient. So then we tried kind of some subtle adjustments to our methods with kind of dynamic thresholds. And I won't spend time on those details now, but we were able to sort of barely achieve this expert level of performance with 30 people. Still way too many people. So how do we fix this? The first question is, why is this happening? So let's do an experiment. So we have answers from 2 different people here. So you might look at this, and so you have one volunteer who put dots where the yellow ones are, and then another volunteer who put dots where the green ones are. And the algorithm doesn't see the megatile underneath. We're not using computer vision or anything fancy like that. We're just looking at the distances. So do you think these 2 dots are pointing at the same object? I mean, they're pretty close together, they might be. Do you think this other yellow one is pointing to the same object as those 2? Probably not, because a person isn't supposed to click twice on the same object. So the algorithm would say, no, if there's a yellow dot there, then this must be a different object. And then what about this one? Well, this is far away. That must be a completely different object. So this is the underlying megatile. And we can see that in this carefully chosen example, the algorithm would get it completely wrong, right? Because this is how it would work. It would say, okay, those are within the distance threshold. We'll combine those. These are within the distance threshold, but there are 2 yellow ones, so we won't combine those. And these are too far apart. So this would be the crowd answer, and the crowd answer would be completely wrong. So when you only have a few people, this is the kind of thing that happens. If you get enough people involved, it kind of comes out in the wash. You have enough people sort of clicking in the similar areas that we see with 50 people, okay, we finally get a really good answer. But it's so inefficient, we were hoping to do something else. So my favorite thing about human computation is that cheating ends up being a good thing. So, most people think cheating is bad. In human computation, cheating is a good thing. And that's because what we do is when we realize that the machine can't do part of the analysis, we just cheat and we put a human in the loop in that one spot. So that's what we're intending to do here. Now, we haven't done this yet, but I'm going to describe to you what we're intending to do. So the idea is that at each collapsing step, we're going to ask the person to make the determination. So if you imagine this is the pseudo code, the algorithm we use to do the megatile collapsing. Here in orange, you can see that's the computer using the distance threshold to decide. So what we're going to do is we're just going to replace those lines of code with a human intervention, and we're going to have a person look at the actual megatile and say, are these 2 dots pointing at the same thing? So it might look something like this. In fact, I was going through the slides with our software engineer this morning. He said, oh, so this is what I'm supposed to do? I said, yeah, this is what you're supposed to do. So in this case, human would look at it and say, yeah, obviously you're pointing at two different things. It's easy peasy for a human. And in this case, a human would likely say they're the same thing. Are we going to ask only one human? We might ask two people to do this just to make sure. And so that leaves us with kind of this cost-benefit analysis. So one question is, whenever you introduce something new, are people going to like it? Are people going to spend time doing it? Because we're going to need people to do a lot of these. And related to that, if it's such an easy thing to do, how's the scoring going to work? I mean, it's going to be extremely rare that somebody gets them wrong, but maybe if they're feeling like they're low on points from the other tasks, this is a way to quickly catch up on points. I'm really not sure yet, and I'm open to suggestions. And then finally, and for me this is maybe one of the biggest considerations, is if this means that instead of 50 people, we only need 5 or 10 people to look at the same megatile, how many people are we now adding because we need people to do this other task? So in the end, does the, do the economics work out? Do we actually end up ahead of the game? Thank you.
Okay, any questions?
What if you showed the annotations by other users to the ones who are reviewing the screen? So you get a screen and you can see what the past people have done. Would that help in you clicking on the one that already exists? So you don't have to merge them. You can just say, I agree that that is also one.
Oh, interesting. So I think what you're saying is that it's my turn to look at a megatile, but instead of looking at a blank megatile, I can see all the historical annotations from the other people, and that can inform where I'm going to click. So I think that would be an interesting thing to pilot. I think that the risk there is you're leading the witness, you're adding a bias there, but maybe that's the point, is that you want that bias to be there. So I think it would be an interesting experiment. Thank you. Yes, sir.
I have one very naive question. As far as I know, in Galaxy Zoo or in other systems, you require very short number of participants, right? Well, about 6 or numbers you just mentioned. What do you think that makes? What is different compared to that? I know that you have many, let's say, galaxies all at once here. So I would say, so it's more than one element that you have to analyze. So I don't know if you have an idea.
So, I mean, if I understood your question right, I think you're asking, like, why is it a problem here when it's not a problem on Galaxy Zoo? And I think one aspect of this that's different than Galaxy Zoo is that we're not we're actually combining identification and classification in the same task. So we're not showing you a lesion and then asking you to tell us what it is. We're simultaneously asking you to find it and tell us what it is. And so then we have this, we've created a problem for ourselves, which is, and maybe that's what you're getting at, and the problem is, how do we know that you're referring to the same thing? If we isolated these things first, that would be actually another way to address this issue.
I guess that computer vision can do that. You are right. So.
Yeah, and maybe that would work in this case as well. I'm showing you some very clean examples. Sometimes they're very messy. Yeah. Anything else? Okay, thank you.
Thank you very much.